Welcome back to Acing AP Biology. In this lesson, we will go over the stages of meiosis. Meiosis can be divided into two phases, meiosis 1 and meiosis 2. Besides that, the stages are the same as in mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. In prophase and prometaphase 1, the chromatin condenses into chromosomes composed of two sister chromatids. The nuclear envelope fragments and the nucleoli disappear. The centrioles move apart and microtubules form between them. What is very important to remember about prophase 1 is that crossing over occurs in this stage. Two homologous chromosomes pair up and form a tetrad formation. It is called a tetrad because there are four chromatids in this formation. Synapsis occurs between homologous chromosomes as fragile DNA breaks and are glued back together at the chiasmata. This is called crossing over. In crossing over, reciprocal pieces of DNA are exchanged, and this increases genetic diversity. In prometaphase 1, microtubules begin to attach to the centromere of the chromosomes and move them to the metaphase plate. In metaphase 1, the microtubules are attached to the kinetochore proteins in the chromosomes, and the tetrads are aligned on the metaphase plate. Independent assortment is set up for in this stage, as each homologous pair orients itself independently of the other pairs. In anaphase 1, independent assortment occurs, where homologous chromosomes separate and move towards opposite ends of the cell. Notice that the sister chromatids stay together. You will see that each end of the cell has a mix of maternal and paternal genes as a result from independent assortment and crossing over. Therefore, genetic variation increases. In telophase 1, each end of the cell now has a haploid set of chromosomes, each composed of two sister chromatids attached at the centromere. Keep in mind there are also regions of non-sister chromatids because of crossing over. After meiosis 1 is complete, cytokinesis takes place with the cleavage furrow in animal cells. It is important to remember that no DNA replication occurs before meiosis 2. The second stage of meiosis, meiosis 2, is the same process as mitosis. In prophase 2 and prometaphase 2, the nuclear envelope disintegrates and the nucleoli disappear. The spindle apparatus forms, and the microtubules begin to attach to the kinetochore proteins of the chromosomes. The kinetochore microtubules then begin moving the chromosomes to the metaphase plate. You will notice that in meiosis 2, chromosomes are composed of two sister chromatids with non-sister chromatid regions. This is unlike meiosis 1, where homologous chromosomes came together to form tetrads. In metaphase 2, chromosomes align on the metaphase plate. Notice the difference between metaphase 1 and metaphase 2. In metaphase 2, the chromosomes align on the metaphase plate, just like in mitosis. But recall that in meiosis 1, pairs of homologous chromosomes aligned on the metaphase plate in tetrad formation. This allowed for the number of chromosomes to be divided in half. In anaphase 2, sister chromatids separate and move to opposite ends of the cell. Each end of the cell now has 3 chromosomes. Lastly, telophase 2 and cytokinesis take place, resulting in 4 haploid daughter cells. The 4 daughter cells contain 3 chromosomes. You recall that we started with a cell that had 6 chromosomes. So, the 4 daughter cells have half the number of chromosomes than the parent cell. This is unlike mitosis, because in mitosis, the parent and daughter cells have the same number of chromosomes, whereas in meiosis, the daughter cells have half the amount of chromosomes that the parent cell had. That's a wrap for meiosis and Unit 9. If you have any questions, feel free to comment below. See you next time. For more bio content, hit subscribe.